Dogs. I like dogs. Laminate. Hey, I'm Jeff Miner. I'm past president of ASMB. I organized the meeting in 2021 in St. Louis, Missouri. I've been a member of ASMB since pretty much when it began, although I did miss the original organizing meeting. But I've attended every meeting since about 2004, as far as I can remember. And I've seen it really grow. I've seen so many of the founders of basically our field come through and serve as president and really contribute a lot and win awards. And now, of course, over the last 10 to 15 years, I've seen the younger people come in and start contributing and see a lot of enthusiasm for matrix biology research. I really like to garden. I feel like when I'm gardening, I can think about absolutely nothing except whether there's enough dirt at the roots and how much water the plants need. It really takes my mind away from the day-to-day rigors of being a scientist. And my favorite molecule, I originally said was laminin because that's really how I kind of started in getting into kidney disease research, because the laminin mutant mice that were made by someone else in my postdoc lab turned out to have a kidney disease. It was very rare, but on my own, I was also making a collagen 4 mutation that also turned out to have a kidney disease, Alport syndrome, which is really what I spend most of my time doing. But I certainly have a long love for laminin. My own favorite discovery, yes, I discovered a new laminin chain when I was a postdoc, laminin alpha 5. And that was pretty exciting. There was clearly some evidence in the literature that this chain existed, but no one really knew what it was, though there was an antibody to it, which made it a little bit easier to discover. But it turned out to be really widely expressed in basement membranes and really important for development. And I remember when I made a knockout, the mice didn't make it to birth. They succumbed to various developmental defects in utero. So I thought, well, this will never be really a human disease protein because the affected embryos wouldn't really survive. But it turns out there are missense mutations in laminin alpha 5 that do cause various types of syndromic changes and effects on different organs, due in part to its widespread expression. So I've spent some time analyzing human mutations in laminin alpha 5 very recently that are found in patients that are born and survive and have kidney defects as well as other defects. The most common thing that we do, probably even since I was a postdoc, is immunohistochemistry, looking to see where proteins are deposited, how they change in disease, especially in the kidney. We're constantly looking at deposition of different proteins in basement membranes as well as in the interstitium that are associated with fibrosis and changes in basement membrane architecture. What brought me to the matrix biology field? I'd say it was my choice of a postdoctoral mentor who was recommended to me so highly by my predoctoral mentor and other people in my lab who thought he would be a great fit for me. So I did go with Josh Sains as a postdoctoral mentor, focused on the neuromuscular junction basement membrane, and that's really how I got into matrix. I had really very little experience or knowledge about the extracellular matrix. In some ways, I kind of didn't think it was really that interesting, but I did realize once I started working on it that the possibilities are endless. Yeah, so I've had many people that I admire in the matrix biology field. As a PhD student, I was in the muscle development field, and there were certainly giants in that world who were very inspiring for me. And when I started attending matrix biology meetings and working on matrix, reading the old literature, seeing and meeting the people like Peter Yurchenko and Billy Hudson and others who are really pioneers in the field. Those were not only colleagues and people who challenged me at meetings, but also people who supported my career over the years through ASMB as well as outside ASMB. They should not rely on what they read, because there's a lot of published research that is just not true, due mainly, I think, to using reagents that are not valid or accurate, like antibodies bought from companies to prove a matrix protein is expressed in a particular site. Other advice would be to seek help from the experts. It's something I didn't do as much as I should have when I was younger, and I felt like in part I didn't want to bother people with my own problems and my own issues, but really there's nothing I enjoy much more than helping young people who have little idea of what they're trying to study understand the details and the nuances that I've learned over the years as a scientist. The memories that I'm coming up with about ASMB over the past 20 plus years is the fun had at the banquets and the parties, whether it was wearing a cowboy hat and dancing in Nashville, or dancing in St. Petersburg, and seeing the Dali Museum, which was really wonderful. So ASMB has done a great job of moving around the country, highlighting other cities, many different cities, and providing an enjoyable time for the people who attend the meetings so religiously during the day, but then like to have a good time together once during the week.